Hi everybody, and welcome to this lecture on the applications, advantages and disadvantages of asexual propagation techniques. You've read in Canvas that asexual propagation involves no exchange of genetic material between male and female cells, so all the resulting plants are genetically identical to the parent plant. They're clones, just like all of the Phalaenopsis orchids you can see on the right at Floricultura Pacific in Salinas. Asexual propagation techniques underpin most of the propagation of the thousands of ornamental cultivars and hybrids across the world, most trees and shrubs for fruit production, and herbaceous crops such as strawberries and potatoes. This video lecture introduces you to some of the applications of asexual propagation, its advantages and disadvantages, and much of this material will be reviewed in other modules. So let's start with the applications and advantages. Asexual propagation techniques are essential when we want to reliably reproduce desirable physical characteristics of the parent plant. For example, perhaps we want to reproduce variegated foliage like the golden lime green tones in the leaves of Diamond Heights ceanothus, which you can see on the right. Normally, this species of ceanothus has leaves that are just plain medium to dark green. Other physical characteristics that are reproduced using vegetative propagation methods include a specific growth habit, a different flower color or size, or differently sized leaves. All of these plants will have a cultivar name, which will give you a big clue that that plant should be propagated asexually rather than from seed. The reproduction of desirable traits isn't just limited to physical characteristics that we can see. We may want to reproduce desirable biochemical characteristics, such as a particular THC level in cannabis. Another invisible characteristic might be disease resistance. Vegetative techniques are essential when we're propagating hybrid plants. If we were to grow hybrid plants from seed, the resulting seedlings will exhibit a wide range of genetic variability. Some may appear to be identical to the parent plants, but remember that some of the genetic variability may be cryptic, meaning we can't physically see it. It's hidden. So there's no guarantee that any of the seedlings will be exact clones of the parent plant, even though they might look as if they are. Therefore, we have to use asexual propagation techniques to reproduce hybrid plants like this Corea Wind's Wonder that you can see on the right, which is a variegated sport of the hybrid Corea manii, which is thought to be a hybrid between Corea reflexa and Corea pulchella. Some plants are sterile, meaning they don't produce any seed or don't produce seed that's viable. So seed isn't even a propagation option with these plants, and asexual techniques are the only way to propagate them. An example is the cultivated edible banana. Some plants bloom very rarely, so again, asexual propagation is usually the only way to propagate these plants on a regular basis. A common example is the vine golden pothos, which is a really common houseplant. Another application of asexual propagation techniques is when we want to reproduce predictably a plant of a particular sex. Most angiosperms are bisexual, meaning the flowers contain both male and female reproductive parts. But some plants produce separate male and female flowers on the same plant, and these plants are described as monoecious. Mono means one, and ecious comes from the Greek word for house. So we have two flowers of the opposite sex living in one house. Other plants produce separate male and female flowers on separate plants, and these plants are described as dioecious, meaning two houses. Sometimes the male or the female plant of a species that's dioecious may be more desirable from an ornamental or a practical or a biochemical point of view. In this case, asexual propagation techniques have to be used in order to reproduce a plant of the desired sex. An example of this is the ginkgo tree pictured on the right, which is a popular ornamental tree both in residential gardens and as a street tree. There are several ginkgos planted in the new part of Aptos village and on the Cabrillo Lower campus near Cessnan House. Female ginkgos produce seeds that are messy and slippery and smell pretty nasty. So these aren't great qualities for a street tree. So male ginkgos are in much higher demand than females and have to be propagated asexually from cuttings. Cultivars of ginkgo are propagated using grafting techniques. Another example of a dioecious plant is our California native shrub, Garrya elliptica, the coast silk tassel. The male flowers are much longer and showier than the female flowers, and therefore more desirable from an ornamental point of view. Almost all the coast silk tassels that you'll see in ornamental landscapes are males, which have been propagated asexually from stem cuttings. Flowering plants grown from seed exhibit two stages as they're maturing, a juvenile stage and a mature stage. During the juvenile stage, plants haven't reached sexual maturity, so they're not able to reproduce. They can't produce flowers, which in turn means they can't produce fruit. The juvenile stage may be very short, for example, just a few weeks perhaps in the case of an annual, but it can be as long as 10, 12, or even 16 years. If you're a fruit tree grower, this can be a problem. You don't want to be waiting all that time before your trees are producing a marketable crop of fruit. This may also be a problem for growers of some ornamental trees that are grown for their showy flowers. One way of bypassing the problem of a lengthy juvenile phase is to propagate these plants asexually from cuttings, where the cutting material is taken from trees that are already mature. Alternatively, you can graft mature wood onto a seedling or asexually produced rootstock. Lastly, another advantage of vegetative propagation techniques is that sometimes you may be able to get a plant to a marketable size much more quickly than you could from seed. Examples include some of our California native shrubs, such as coffeeberry, ceanothus, and especially manzanita. It can take several months for manzanita seed to even germinate, let alone grow to a transplantable size. In contrast, a stem cutting is typically around 4 to 5 inches long and will take usually between 12 to 16 weeks to root. So by the time you've got a young manzanita plant that's big enough to transplant from a cutting, you may not even have germination or emergence if you're growing manzanita from seed. So let's look at the disadvantages now. While cloning can be advantageous, the loss of genetic diversity also has disadvantages. For example, it can make commercial crops vulnerable to diseases. And a relatively recent example you may have heard about is bananas. Over 90% of the edible bananas sold worldwide are the same variety, the Cavendish, and the global supply of this variety is threatened by a fungal pathogen which causes the deadly and incurable Panama disease in banana plantations. Another example is the tequila agave in Mexico, which has been threatened by both a bacterium and a fungus, causing the death of many agaves used for the production of tequila. Plant scientists and breeders are working with growers to find practical solutions for these problems. In restoration nurseries, the reduction in genetic diversity that could result from propagating too many plants of a single species asexually could result in a lack of adaptability to environmental changes such as global warming. So if you're going to propagate restoration plants asexually, material should be collected from as many different plants as possible in order to maintain a relatively high level of genetic diversity. Alternatively, rely on seed propagation rather than asexual propagation. So in conclusion, you can see that there are many instances where asexual propagation techniques are required or produce faster results than growing from seed. Your job as a propagator is to select the most appropriate method, always bearing in mind, of course, that there can be disadvantages to asexual propagation techniques, especially as far as the loss of genetic diversity is concerned. That's the end of this lecture. Head back to Canvas now to continue with this module, or go take a break.